I'm a neuroscientist, and I work in a medical school. And in medicine, we like to compartmentalize disciplines. So neuroscientists are supposed to be focused on things from the neck up, predominantly. And indeed, my neuroscience colleagues wonder, John, if you're interested in the neurobiology of emotions, which I am, why do you focus on the gut? Well, maybe we just look at our language. Gut feelings, gut instincts, gutsy moves. We're gutted when we're disappointed. We have butterflies in our tummy when we're nervous. So perhaps there is something really to these phrases, and that we do personify these emotions deep within our gut. And over the last decade or more, our research group in Cork, in the very south of Ireland, has been focused not only on how the gut speaks to the brain, but even more how the trillions of microbes that reside within each of your gut, how they play a role in programming all aspects of health, including brain health. And this field is really exciting and really new, and it's taken me and our research group on all sorts of journeys that we never expected. But like most things in modern day medicine, perhaps they're not that new at all. Here is Eli Metchnikoff. Metchnikoff won the Nobel Prize in 1908. He's one of the fathers of modern immunology. He discovered how immune cells eat up things that they're afraid of. And he was Russian, working at the Pasteur Institute in Paris. And as often happens, great scientists, later on in their career, they can start coming up with crazy ideas. And Metchnikoff was full of such crazy ideas, many of which never held up. But one of his ideas, and one of the things that he focused on towards the end of his life, was why did people in parts of what is now rural Bulgaria, why do they live longer, happier lives? And he wrote about it. And he looked at some of the factors. And what he put it down to predominantly was what they ate. And what they ate was a lot of fermented foods containing lactic acid bacteria. And so that was over 100 years ago, but a lot of his ideas laid in abeyance for at least 70 or 80 years. Now, my research group is really interested in how stress affects the brain, but also how stress affects the entire body. And I'm talking about chronic stress and traumatic stress. And we're particularly interested at stress at key epochs across the lifespan. Stress in the perinatal period, stress in adolescence, and stress in old age. And a number of years ago, using an animal model, we found that animals that were separated from their moms from postnatal day 2 to 12, right early in life, that they developed a whole body stress response. Not just affecting what was going on in their brain, but there were marked effects in their brain, affecting their pain responses, affecting their immune system, even affecting their barrier function in their gut. But one of the things we found, almost by accident, was that there was a change in the composition in the microbes in the gut. That there was a reduction in what we would call diversity. That there was a signature of this early life trauma that persisted into adulthood. Now that could be epiphenomenological, but it got us thinking, well, maybe that, by stress affecting the microbiome, this might play a role in our thinking about the microbiome. So what is this microbiome? Well, a few figures for you. First of all, we are living in a microbial world. The microbes were there first. And there's never been a time where the brain has existed without microbial signals. Some key statistics, as a neuroscientist, it's quite humbling to think that our microbiome basically weighs the same as our brain. It's about 3 pounds. And if we talked about all of the genes that we have, we are more than 99% microbial. That's humbling. Think of all the money we spent on the Human Genome Project, and it's less than 1% of our genes. If I'd given this talk a couple of years ago, I would have said to you, we have 10 times more microbial cells than human cells. But this is a very fast field with advances in sequencing technology and algorithms all the time. And so now that's been revised downwards to about 1.3 to 1. So we're still more microbial, but just about. So the next time you go to the bathroom and you get rid of some of your microbes, just think you're becoming more human and what that may mean. Where does it all begin? Where do we get these microbes? Well, we're taught for the most part to be sterile in utero. And as we are being born, as we emerge through the birth canal, we get these early microbes from our moms, and they colonize the gut and inform the immune system and inform the rest of the body. So therefore, a lot of factors early in life can influence the composition of these microbes. Not least of all is the mode of birth, mode of delivery at birth. So if you're born by C-section, you bypass this, what I would call a microbial baton in a relay of life from our moms. And we know that with escalating C-section rates in the developing world and throughout many countries, that there is an increased relative risk of asthma, allergy, and type 1 diabetes. And we know very little about what the impact is on our brain health and our stress response. And this is something that we're studying avidly in Cork at the moment. I poo-pooed genetics a little while ago, but genetics are really important here. Host genetics and microbe interactions is probably one of the biggest areas for understanding inter-individual differences in both the microbiome composition and in the response to interventions, and will be also something that will gain a lot of attention in the times to come. Hospitalization, of course, antibiotics will influence it. The environment we live in. I mentioned maternal stress, but also maternal infection and gestational age. One of the key things also is mode of nutritional provision. Human breast milk has the highest complexity of sugars of all mammals. These human milk oligosaccharides, the intriguing thing for me about them is that they're not digested by the infant at all. They haven't got the ability, but they're digested by the microbes. And these microbes turn these sugars into key chemicals that support brain development, for example, like sialic acid. Switching to old age, I want to highlight a study from my colleagues in Cork that was published in Nature a few years ago. And they took almost 200 elderly individuals and looked at a variety of different health indices, including frailty, cognitive function, etc. And what they found was those that were more healthy had more diverse microbiome. And that was really key paper at the time. But they went one step further to look at what was driving this diversity. And what they found was those that had a diverse diet that changed regularly and that lived in the community had a much better health outcome than those that often were in nursing homes having a lot of processed food. So again, we not only have a gut-brain axis, we have a microbiome, gut-brain axis, but now we really begin to reinforce that it's a diet that is one of the key drivers and regulating this microbiome. And we're only beginning to really understand slowly how these diet and microbe can interact with our body system to promote health. What we do know is, as I alluded to, these microbes are little factories producing all kinds of weird and wonderful chemicals that support all aspects of our health. And we're beginning to slowly tease apart what these are and how they influence all parts of our body, including our brain. And as a neuroscientist, this is something that I've been avidly working on. But how do we study it? In biology and in engineering in general, probably the easiest way to study something is take it out and see what happens. So we have the ability to do this in mice, and so we've basically created germ-free mice that lack any bacteria. And they have no, perhaps, human translation with the exception of the boy in the bubble. These are the mice in the bubble. But they allow us to answer the question, is the microbiome involved, yes or no? And through this strategy, we've been able to find things that we never expected. First of all, we've been able to show that the microbiome is regulating myelination. Now, myelin is the insulation on which nerve cells communicate, and we found that this was disturbed in these germ-free animals. It also influences the birth of new neurons, a process called neurogenesis, changes in neurotransmitter levels and plasticity in the brain, and also in terms of dendrite growth. Microglia are among the brain's immune cells and how they become activated. And even the blood-brain barrier, which protects our brain from all things in the outside, is changed in these germ-free animals. And you don't need to be a neuroscientist to be able to identify in this figure that this is a neuron from a germ-free animal compared to control. And this tells us that for normal neurodevelopmental trajectories, we need appropriate microbes within our gut, and that this leads to aberrant behaviors relevant to anxiety, to fear learning, to autism, and to the stress response. And this has been a really exciting journey. It also means something else, because if stress is affecting the microbiome and the microbiome is affecting the stress response, as we've shown in germ-free, could we now target the microbiome to alleviate some of the effects of stress? And this leads us to the concept of psychobiotics. It's a term my colleague Ted Dinan in Cork came up with a few years ago, and it really just means targeting the microbiome for mental health benefits. Initially, we started in animals to prove that certain strains of bacteria, as well as prebiotics, which are dietary influences, could indeed reduce anxiety, change cognition, and reduce the stress response. Moreover, they could modulate neurotransmitter levels within the brain. We've now gone on and done the exact same in healthy volunteers in humans, and been able to show that some of these specific interventions are also able to dampen down the stress response in healthy humans. And we stress our humans by putting them through public speaking, like what I'm going through right now in the laboratory, or also putting them through exams, which is what we do a lot of in universities. And we find that those that take some of these interventions have a lower stress. But the question is how? How do bacteria signal to the brain? And we have a nerve called the vagus nerve, which is a long wandering nerve which communicates from our brain to our periphery and from our periphery to our brain. And a number of years ago, together with John Bienenstock's group in Canada, we performed vagotomy in mice to see whether the long-term effects of a lactobacillus bacterial strain that reduced the anxiety and stress response were affected. And indeed, they were completely gone when we cut off the vagus signaling. So this tells us that what happens in vagus doesn't just stay in vagus, but will actually affect our emotions. And then this is one key pathway for how microbes could signal to the brain. This is really, we're at the beginning of this journey, and it's really exciting and really new and transforming how we conceptualize treating psychiatric illness and enhancing well-being and enhancing happiness in general. And it's so new, except, of course, as I already told you, there's very little new in this field. And if we had read the British Journal of Psychiatry from 1910, it would have told us already about treating melancholia with lactic acid bacteria. So perhaps we are just going back to the future. So what we have now is the evidence to support the concept of psychobiotics, that we're at the beginning, as what we've said is a psychobiotic revolution. It's just the beginning, but it really is able to interface where we are between mood and food, and how diet gives a scientific rigor to the concept of you are what you eat and you feel what you eat. There are things we can offer. People always ask me, how do you try and have a happier psychobiotic life? What can we do to enhance this? Of course, one of the key things is increasing your intake of foods that really support your microbiome. These include prebiotics, which are generally fibers and vegetables such as artichokes and leeks and onions. Taking omega-3 fatty acids. Omega-3 fatty acids support the development of the microbiome. Polyphenols. Now, polyphenol might sound like a 70s disco queen, but polyphenols are present in a lot of foods, including nuts, seeds, coffee, dark chocolates, so some good foods. They're also in red wine that people talk about, but I should also say it's equally in grape juice. So polyphenols have been shown to also promote the development of beneficial bacteria in your gut. We know, as I mentioned from Metchnikoff, the fermented foods and yogurts are also very positive towards supporting your microbiome. In terms of what you should not do, well, you should try and decrease the intake of processed foods. Ireland, just behind the UK and Germany, has one of the highest processed food intakes in all of Europe. And we know that processed food intake it has negative effects on mental health. And there's direct evidence supporting emulsifiers and sweeteners negatively impacting the microbiome. And maybe some of the negative effects of processed food is due to its effects on the microbiome. A lot of new data supporting how exercise affects the microbiome in a very positive way. And of course, it's easy to say, but we should try and avoid stress and avoid sleep disturbances and jet lag, all of which are shown to be negatively influence the microbiome. We need to try and avoid C-section as much as possible. However, I do want to say that C-section is a medically indicated life-saving intervention, so you can only minimize it to a certain level and then support breastfeeding as much as possible and minimize antibiotic use. One of the intriguing things is also having a pet will also increase diversity. The last thing I want to allude to is, because this is a global forum, and the microbiome field is truly a global field where people are looking at it across cultures. And if you go to rural Tanzania, where the Hadza community are still hunter-gatherers today, and look at their microbiome, it's very diverse. And this correlates to their diet of rich in fibers. We can chart the introduction of agriculture, which is now seen in rural Venezuela and Malawi, and you can see that in these populations there begins to be a diminution of the microbiome. But in our Western world, with the advent of processed food, increased stress, antibiotics, both through medication and through the food chain, we are actually extinguishing components of our microbiome, and that this is really a dangerous part we are. We know in these communities here, there is no chronic inflammatory diseases like multiple sclerosis and inflammatory bowel disease. We don't know much about it in terms of mental health disorders, but it's something that we have to appreciate overall. Our state of mind is directly influenced by our state of gut. So Hippocrates is the father of all medicine, and he said, let food be thy medicine. I'll just paraphrase that a little bit more and perhaps say that maybe let food for your microbes be thy medicine. And I hope that's given you some food for thought. Thank you very much.